like what germ layer forms the nervous system, Okay, let's talk about nervous system embryology. And as we do, we're going to answer a few questions like what germ layer forms the nervous system, what is neurulation, and what are neuropores and why are they clinically significant? Let's start with an overview to see some of the main events that occur in the development of the nervous system. To start, in week 3, we have the development of the trilaminar embryonic disc. And from that disc, we're going to have the development and the formation of a neural tube in week 4. And it's from that neural tube that we're going to have most of the nervous system developing. Then in weeks 4 through 6, these different vesicles are going to form in the cranial end of the tube, and that will eventually become the brain. And then finally, we know at birth, we're going to have the fully formed nervous system. So that is our overview. Primarily in this video, we're going to talk about early embryonic development through the formation of a neural tube. And I know it's been a while, so we're going to do a rapid embryology review to get to the trilaminar embryonic disc. In week 1, fertilization occurs, and we eventually get to this 2-cell stage. And then from that stage, those cells are going to continue to divide until we have this structure that looks kind of like a mulberry. And so we call it a morula because that means mulberry. And then from the morula, eventually we're going to get to this stage where we have a blastocyst. So some of the week 1 highlights, the oocyte is fertilized by a sperm, typically in the ampulla of the uterine tube. And then a diploid cell with 46 chromosomes, that's what we call a zygote, is formed. And then the zygote divides and eventually forms that blastocyst that we see here. Moving on to week 2, the blastocyst completes its implantation into the endometrium of the uterus. So looking at this image here, we see this is the endometrial lining. The blastocyst that we have here is now implanted into that lining. And then a bilaminar embryonic disc is now formed. So if we look here, we see we have this epiblast layer, then this hypoblast layer. So those 2 layers are what make up the bilaminar embryonic disc. Now in week 3, we still have that bilaminar embryonic disc, but as we continue forward, we see this primitive streak develops, and it looks like we have this layer that's starting to develop in between our initial 2 layers. And if we're to zoom in here, we can see we now have this pinkish layer that's kind of almost involuting in, and we now have 3 different layers. And so this process is called gastrulation, and that is taking the bilaminar embryonic disc and then converting it into a trilaminar disc, tri meaning 3, so it now has 3 germ layers. So I remember that week 2, bilaminar, 2 layers, week 3, trilaminar with the 3 layers. Let's take a closer look at the trilaminar disc. We're going to label some of its components and see what role they play in the development of the nervous system. So the first layer we're going to label here is this outer layer in blue. That is the ectoderm. Ecto just means external or on the outside. And then in yellow here within is the endoderm. And that's a great name because endo means internal or within. And then in the middle here, a lot of this pinkish that we're seeing is the mesoderm, and meso just means middle. So we're going to label some of those mesoderm components. The first being this paraxial mesoderm here, and it just gets this name because it's next to the axis. And then this portion in the middle, we're going to call the intermediate mesoderm. And then the most lateral portion, we're going to call it the lateral mesoderm. So we've got some really good names there. And then the lateral mesoderm has 2 different components to it. So it has a splanchnic portion and then a somatic portion. Finally, we're also going to label the notochord right here. And the notochord actually plays a really important role in the development of the nervous system. It is actually going to induce a part of the ectoderm to differentiate and become a specialized type called neuroectoderm. And to get a better idea of that, we're going to look at this new image here. So this is a dorsal view of an embryo at day 17. And we can see this is the notochordal process right here. And you can see that this area surrounding the notochordal process is now called the neural plate. And again, the neural plate is just the specialized ectoderm that has differentiated and is now neuroectoderm. Since the notochord has induced this ectoderm to differentiate and form the neural plate, we're now going to zoom in here and follow the components of the neural plate through time. Starting out here, again, we have the notochord, but then you're starting to see we get kind of this pit and these folds going on. So this darker blue section we're going to call the neural fold. And then this pit that we now have created is going to be called the neural groove. And then this ridge or this crest right at the edge here, the peak, we're going to call that the neural crest. And we see we've got that on either side. As we continue to move forward, we see that these neural folds here are coming closer to each other. We still have a neural groove or that pit and then a neural crest on the ridge. Moving even further, the neural folds are getting closer. We still have a neural groove, and then the neural crest is kind of starting to bud out a little bit. And then still in the light blue, it's kind of been throughout, but we have the surface ectoderm. Moving from image D to the image E, we've had some serious movement going on here. The neural folds have finally met, and we're no longer going to call this neural folds because it's now a neural tube. They've met and connected. And instead of calling it a neural groove now, this section in the center, we're going to call a neural canal. And then we have the neural crest cells kind of right just external to the neural tube. And then the ectoderm is now met, and we have surface ectoderm most externally. Finishing up, the neural crest, it was kind of combined in the center, but as we move forward, we now get these separated components of neural crest. So this process we just went through is called neurulation, and it's the process of forming a neural tube that's covered by ectoderm. And one note I have on this, until fusion of the neural fold is complete, there are going to be openings in different ends of the neural tube. So we're going to look at that on this image here. And so this is my little PowerPoint creation of a closed tube. So this is eventually what we want with the nervous system, a closed tube. But in development, we have these different openings. So the neural folds fuse, you can kind of think in the middle is where they start to fuse and they almost zipper on the ends. So this yellow portion would be openings of that neural tube. So you can see we have an opening on top. We're going to call that the rostral or the cranial opening. And then we also have an opening inferior, and we're going to call that the caudal opening towards the tail. Looking at that on an actual embryo, we see we've got a dorsal view here. So it'd be the cranial end. We've got the caudal end. And then the neural tube would be fusing. It's kind of fused in the middle. but we have these openings. So we have a rostral opening, and we also have a caudal opening here where the neural tube is yet to fuse. Instead of calling these openings, we're going to give them a little bit more of a specialized name. So this rostral one, we're going to call a rostral neuropore. And a pore is just a passageway or an opening in neuro because it's in the neural tube. And then we also have a caudal neuropore. And so these neuropores, they continue to fuse as we develop. So we see a look at day 23, there's a little bit more fusion that has occurred. And then even at day 24, there's more fusion until they're eventually completely fused. So a recap on these, neuropores are openings or passageways in the neural tube. The rostral neuropore closes on day 25 or around there, and the caudal neuropore closes around day 27. And one note on these, maternal folate deficiency is associated with failure of the neural tube to close. And failure of either of these neuropores to close will result in what are called neural tube defects. And we're going to have a video that goes through those in a little bit more detail, but there is pretty high clinical significance with these neuropores. And that is it for this introduction to nervous system embryology. So let's do a quick review. We learned the nervous system is derived from the ectoderm that we can see here in blue. And specifically, the central nervous system is derived from the neural tube. And then most of the peripheral nervous system and autonomic nervous system are derived from neural crest. We also learned the process of neurulation. And in that, the notochord induces the ectoderm to differentiate into neuroectoderm, which forms the neural plate. So notochord forming the neural plate. And then the neural groove, neural folds, and neural crests appear in the neural plate. And we can see those well here with the neural folds, neural crest, and then the neural groove, or this pit right here. And then eventually those neural folds are going to fuse and form the neural tube, which we're seeing here, fusion, a neural tube, and then the neural crest on either side. Then finally, we learned about neuropores. And neuropores are openings at the ends of the neural tube. So we've got a rostral neuropore here at the cranial end, and then a caudal neuropore. The rostral neuropore closes around day 25. The caudal neuropore closes on or around day 27. And then maternal folate deficiency is associated with failure of the neural tube to close. And that is it for this video on nervous system embryology.